Okay, so in yesterday's lecture, I did speak about Drosophila gastrulation or mesoderm invagination. And I tried to focus on how gene regulatory networks that are triggered by the gradients initiated as part of the dorsoventral patterning system actually lead to effects on cell behavior in the mesoderm. So the focus of what I was trying to tell you yesterday was to understand how gradients along a particular axis actually become transduced to bring about the dynamics of the tissue. And in this particular case, we were looking at how the nuclear dorsal gradient facilitates the invagination of mesodermal cells on the ventral side of the embryo. The focus of today's talk will be to think about three different kinds of tissue sculpting, namely bending a tissue, of which ventral furrow formation is an example, tissue elongation, and you could say tissue or cell sheet fusion. I'm going to use these three paradigms to try and tell you how you can get tissues to acquire different forms. I will also point out that all of these three morphogenetic transformations are associated with single layers of cells, and in tomorrow's class we will get more complex and deal with morphogenesis that involves more than one cell layer. So as we saw already yesterday, in order to be able to bend a tissue, you need to constrict the okay, so in order to be able to generate the kind of curvature you saw in the previous slide, right, which you can sort of call a negative curvature or a buckling of the tissue so that you have a negative curvature here, you need to be able to constrict the surfaces that are facing this way, which we call the apical end of the cells. And we saw that that apical constriction relies on actomyosin complexes that are located medially at the apical end of the cell. We also saw that these complexes contract in a way that's pulsatile. So we spoke of a sort of stepped pattern of constriction that was associated with phases of contraction followed by phases of stabilization. We saw that this constriction was associated with the apical accumulation, actomyosin complexes that contracted where you could see contraction of the actomyosin network and deformation of the membrane, especially at points at which this meshwork was connected to the adhesion molecules on the membrane. We said that this sort of constriction functions like a ratchet. And one of the reasons why this sort of constriction may have a specific utility is that they have a higher probability of having neighbor cells also contract. They also help buffer contractions within a sheet and prevent competition between neighboring contractions to somehow ensure that you have collective behavior and that no single cell can sort of cue or destroy the collectivity of the behavior within the sheet. We also discussed yesterday that in order to achieve these these kinds of apical constrictions, you need to distribute not only adhesion proteins, but also regulators of the actomyosin cytoskeleton. And this just shows you how an activator of the pro-GTPases also becomes enriched apically during the course of these constrictions. Then we saw that in mutations in genes that are part of this gene regulatory network, that redistribution of ROGEF2 that you saw in the previous slide does not occur normally. So in essence, what we need to be able to do to achieve this constriction is to ensure that you facilitate apical constriction of this ratcheted kind, specifically in the apical region of these cells of the mesoderm. And we also saw how the two transcription factors that were the targets of that dorsoventral fattening gradient have distinct effects on adhesion and contractility. So in some sense, we could say that what apical constriction generates for a tissue is a sort of negative spontaneous curvature of the cell layer, which buckles inwards. What I've just described to you are the cellular and molecular mechanisms that allow this to happen in a collective way so that a strip of cells coordinately invaginates and gets internalized. We also saw that in addition to this gene regulatory network, which offers some kind of top-down control, there was also experimental evidence that suggested that you could modulate this process mechanically. And I showed you some experiments that showed that if you apply force on embryo using either magnetic tweezers or a micropipette, you could actually rescue or reinstate gene expression that had been lost in genetically perturbed embryos, suggesting that in this process, mechanical cues can trigger gene regulatory network, right? And one implication of this was that maybe in the course of normal development, mechanical forces induced or generated by other tissue deformations could act as triggers for gene expression, right? Okay, so having described some of this yesterday, I will now sort of move to looking at a different kind of morphogenetic event that primarily accomplishes elongation of the tissue. So this again is an example from the Drosophila embryo, and what you're seeing is the Drosophila embryo at progressive stages in its development. And all of what's labeled here in purple is a structure we call the germ band. For simplicity's sake, just think of it as the sheet that as part of the sheet that surrounds the whole embryo, right?
So when you say stage 6, 7, 8, it's not nuclear division.
When I say what?
Stage 6, 7, 8, it's not the nuclear division cycles. So it's the divisions
You've
have
already got a lot of morphogenesis that's already happened. It's cellularized. The pole cells are getting internalized. There are these paros. So, but this broad purple shaped structure progressively becomes narrower and longer and adopts this sort of U-shaped configuration. This structure, we call this structure the germ band. And if I had to use the same sort of sections that you saw in the earlier slide, this is the mesoderm. So really all of this bit here is what we might call the germ band, right, in cross section. And how does this elongation come about? Before I go to that, let's just go back to looking at this movie. So this is where the germ band is extending. So it came all the way up to this point where you have these big bright cells that somebody asked me about, right? Prior to that, it was sort of tucked away here. And after that, it's again going to make its way backwards end up sort of being roughly where it was before, right? So the first part of what I showed you, I'll play it again, was that germ band, which was just restricted to this part of the embryo, sort of moves forwards by elongation and ends up with the second stem of the U in this part of the embryo. And then after that, it makes its way back and returns to the posterior or tail end of the embryo. I'm just going to play it one more time so you so that's where the germ band is extending and making its way forward. Good, right? And it stalls for a little bit before it starts moving, let's say, collectively again in the reverse direction, so that this front now makes its way back and ends up where it was. So the first part of what I showed you, we call germ band extension. Second part of what I showed you, which is the reverse process, we call germ band retraction, right? So much of what I'm going to show you now is the process of germ band extension. So really we are following this front here, and you'll see that this structure that's labeled AS is the amnioserosa. That's the structure that you saw had the big bright cells. And what you're progressively seeing is that this front here is moving in this direction, right? And moves all the way to the region of the head. This is the event that we call germ band extension. And if you look at what it achieves, it converts a tissue whose shape and dimensions were like this into one that's narrower and longer, right? And to obviously fit, it's sort of folded upon itself into that U-shaped structure. So really what this is achieving is, what's happening is that the tissue is kind of converging in one axis and extending along the other axis to change the aspect ratio. So the question is, how can a tissue change its dimensions from this to this? And at least one way that people found this could happen was by changing the way cells within this broad band interact with each other. So what you'll see in this cartoon is that if you have a row of rows and columns of cells that are arranged like this, most cells in this tissue at this stage are roughly hexagonal, let's say, on their apical surfaces. Essentially, what happens is that cells now exchange their neighbors so that the linear arrangement that you saw previously is now disturbed in this sort of way. So you could also simply, if you just had a strip of 2 cells, for example, or 2 columns of cells, let's say one here and one here, you could convert that into a single column of cells just by getting cells in each column to move towards each other so that the arrangements you would have would start off to be like that. So they're sort of moving towards each other till you just end up having arrangement like that. So what this kind of process requires is that the cells in each column have some sort of directional mobility of the kind that we saw in the hemocyte, right? So that this cell will now put out some protrusions so that it can move a little bit in this way. And this cell, on the other hand, will now put out some protrusions so it can move this way till you have them sitting one on top of the other, like that. So if I had a 2-cell wide column, I could now make a twice as long 1-cell column, where instead of cell 1 and 2 being oriented like this, I'll have 1 and 2 being oriented like this, right? So in effect, what you have to do to achieve this kind of thing is to polarize one end of the cell differently on the 2 sides so that they can migrate within quotes in opposite directions. That's one way of thinking about how you can do this. This is another way by which you can do this, and this is what happens in this process that I just spoke about. And that is to actually take one of these interfaces at the point of that's formed at the point of contact to adjacent cells and shrink that interface to a point so that now the cells become oriented in this configuration, right? At this point, you will see that these cells still remain neighbors, but these cells have now become neighbors. Initially, these two were separated from each other. You now grow back this interface in the opposite direction, and you actually separate these 2 cells so that they're no longer neighbors, and these 2 cells that weren't neighbors before become neighbors. So you could say some cells gain neighbors, some cells lose neighbors, and you've sort of jumbled up this configuration to something that looks like this, simply by directionally altering the length of an interface between 2 cells. But in order for this sort of mechanism to give you reliable change in the dimensions of the tissue, you will need a mechanism by which, let's say, all interfaces that are oriented in this direction shrink, and then they grow back in the opposite orientation. So if I have a field of cells that looks like this, I'll need to say that all interfaces that are oriented along this axis, which is the dorsoventral axis of the embryo, the same axis we discussed in this example, all the interfaces between cells that are oriented along this axis need to shrink. All interfaces that are, and then after they've shrunk, they need to grow back along the AP axis. So in other words, you could say that you can achieve neighbor exchange also by directing by directional shrinkage of cellular interface. You could also think about doing this in a more efficient way than just 2 cells at a time, by somehow getting these cells that are now arranged in an interlocked manner so that they're, cells in one row are a little bit staggered from cells in the other row. You can get cells to form columns by aligning one set of interfaces like this. And then you could treat this whole long linear structure as if it were one interface, and then shrink that whole interface to a point so that you get a structure that looks like this, which doesn't have just 4 cells, but actually lots more cells. And then you start growing interfaces in the opposite direction. So you're achieving the same thing, but you're doing it many cells at a time, right? So this requires you to first orient or align these cells such that the interfaces form a column, and then shrink that line till it becomes a point, and then grow it in the opposite direction. And this is actually what has been nicely visualized by the labs of Thomas Lequi and Jen Zalen. And if this movie will play now. You will see that you're looking at this field of cells, and you're looking at how this field of cells actually changes its morphology to become longer. And you would have seen in the process that cells that were initially neighbors cease to be neighbors, right? So if you look at the yellow, blue, and magenta, they're actually different rows of cells, and you will see that over time there's mixing of cells and an exchange of neighbors. In some parts of it, it looks a little bit like this, where you have single cells adjacent to each other. So a question that you can then, so the way people actually discovered this was to actually track cells within the sheet and look after a period of time where those cells ended up. They also looked at the frequency distributions of these morphologies of the interfaces, which, as I showed you in the previous slide, go from being like this to being X-shaped and then resolving to this opposite direction. So you can call them type 1, type 2, type 3, and you can quantify the frequency at which each of these morphologies occurs at some time points during this process. What you will see is that as this movement proceeds, you go from having an abundance of type 1 interface morphologies to an abundance of type 3 morphologies through an intermediate stage. So these sort of morphological transitions are called T1 transitions, and you will hear in the main course, possibly both from Frank Ulischer and Guillaume Salbreux, some of the physics and the models for vertex models for these transitions. Okay, and this is just an example of a real life multicellular rosette where cells align themselves into columns and then form a rosette-like structure that then resolves in the opposite direction, allow extension in the opposite direction. What you're seeing here is the distribution of two proteins, right? One in red and one in green. And what you can appreciate is that over time, you will see that that red protein is enriched along interfaces that are oriented primarily along this axis, which is the DV axis, right? And the green protein is primarily enriched along the interfaces that are oriented in the AP axis. So what this tells you is that there are proteins whose distribution in a cell is dependent on the orientation of that interface along the embryonic axis. And so a question that arises is whether these distributions have are of consequence for sort of transforming the junction morphologies along the lines that you see here. So again, what people have been able to show is that at specific developmental stages, you can see very pronounced alignment of these interfaces. And these interfaces also become enriched in this particular protein here, which is essentially myosin. What I'm going to now show you is a summary of what people have seen with respect to what happens to proteins during these transformations. And here we're just looking at the shrinkage of these DV-oriented junctions. What people have found is that myosin becomes enriched at the shrinking junctions relative to the non-shrinking junctions. What people have also seen is that this adhesion molecule, E-cadherin, becomes, or its levels reduce at the shrinking junction by a process called endocytosis. So you could say that these shrinking junctions are characterized by an enrichment of myosin and a reduction in E-cadherin, right? What people also saw, and this is the image I showed you in the previous slide, was that myosin is enriched along these interfaces. There is a polarity protein, the same protein that's also polarized to the leading edge of a migrating cell, is enriched along oppositely oriented interfaces. And when you have cells exchange neighbors in these multicellular columns, again, you see that as these interfaces shrink, they accumulate actin and myosin, and they have a relative reduction in E-cadherin. But as this point sort of begins to grow to form new interfaces, they accumulate E-cadherin, but also this protein here in green, which is bazooka. People have then gone on to show that some of these proteins, for example, bazooka or the regulation of myosin activity, are essential for mediating these junctional transformations. So you could say that it's essential to distribute the cytoskeleton, adhesion, and polarity determinants in a polarized manner at these interfaces for you to be able to achieve the systematic shrinkage of a large band of cells so that you can achieve the deformation of the tissue, right? So I also just want to say that these interfaces that are oriented along the dorsoventral or AP axes are mechanically polarized. What do I mean by that? What I essentially mean is that if you could measure the tension at each type of interface, you would find that the tension at the DV-oriented interfaces is different from the tension at the AP-oriented interface. So how would you measure that? You would measure that in the kinds of experiments I'll show you here, which is to use a laser to make a nanometer size nick in one of these interfaces. And then just like you could cut a rubber band, you would look at how quickly the cut ends actually recoiled to give you a sense of the tension at those interfaces. And essentially what people found by doing experiments of this kind, which you'll see here, it's a very short movie. By the time I come there, okay, start again. Right, so that's the interface that was cut. And you will see that the two ends actually retracted from each other. So these were DV-oriented interfaces. And now here, this is an AP-oriented interface, which was cut. And you can look at how quickly, or what's the velocity of their recoil is immediately after the cut, and get an inference of the tension. And such experiments have revealed that the tensions at the two interfaces are different. And again, going back to mechanics, people have also shown that you can polarize myosin distribution by simply applying an external mechanical stress. So if you press on the region of the germ band using a micropipette, that's enough for you to recruit myosin tag to GFP to those points. So you could say that not only are the dorsoventral and anterior-posterior oriented interfaces different with respect to their protein composition, they're also different with respect to tensions at those interfaces. And other experiments indicate that you can recruit myosin simply by applying a mechanical force. And so you could now begin to ask, okay, all of these distributions exist, these polarities exist. How do these polarities come about in the first place? And to look at that, we have to go back to the patterning system that Vijay briefly discussed, and I just briefly mentioned, that which is the anterior-posterior axis patterning system, which we discussed is initiated by gradients that emanate, two gradients that emanate from the anterior and posterior poles of the embryo to govern gene expression patterns. First, subdividing the embryo into broad domains of gene expression, and then hierarchically subdividing those domains into narrower domains of gene expression. One class of proteins that appears to be regulated by this segmentation gene network are a set of proteins that are called TOL. And if you remember, TOL is also the receptor that binds the ligand to initiate the dorsoventral patterning cascade. But what I will show you is that there isn't just one TOL protein, but there are the family of TOL proteins that appear to be expressed in a stripy pattern along the anterior posterior axis. So you can see there's a tall 2, the tall 6, and tall 8, and they all make stripes. But you can look at how these stripes relate each other. And what this diagram actually tells you is that they're expressed in overlapping patterns, so that would argue that each column of cells within this germ band might have a unique tall code, meaning some combination of high and low of different tall receptors. What you'll also see is that these stalls nicely mark, so they're expressed on the membranes of the cells of the germ band. And in particular, experimental evidence showed that if you take away these 3 tall receptors from the embryo, you fail to polarize the interfaces along DV and AP axes. So these proteins are all necessary to achieve the polarization, so the polarization in molecular terms of the interfaces along the AP and DV axes. What's also true is that the genes that are whose expression depends on these gradients in the domains in which of the germ band which is actually extending also govern this planar polarization of cellular interfaces. So if you knock down genes like Runt and Kruppel, which are sort of next in the hierarchy after these maternal gradients, they also disrupt this polarization. So what that tells you is that the gene regulatory network that operates downstream of the gradients along the anterior posterior axis govern the polarization of the interfaces. And at least one set of membrane-associated proteins that are critical for this appear to be the family of tall receptors. Right? The other things we've looked at so far are actually transcription factors. The tall receptors are proteins that are receptors on the membrane. So they're more likely to have a structural role, and there's evidence now about how they function. I've not sort of included that here. So the next thing I want to come back to, so we've said that there's this gene regulatory network that governs the polarized remodeling of cellular interfaces. We've said that these interfaces exhibit both molecular and mechanical polarization. We discussed how the Rho GTPases are key regulators of actomyosin contractility. And in this piece of work from Thomas Lecui's lab, they basically, and we also discussed pacemakers, how proteins that regulate the Rho GTPase switch can act as pacemakers. So what I want to also say is that the enrichment of myosin along these interfaces that are oriented the DV axis results from actomyosin flows towards these interfaces. So the flows actually destabilize the interface, but the enrichment of myosin that occurs as a consequence of these flows actually stabilizes these interfaces. So what this work actually argued, so like someone was asking me yesterday, so typically we would say that the Rho GTPases govern through their downstream effectors like rho kinase the activity or they help generate active contractility through the actomyosin cytoskeleton, right? What work here showed was that properties of the actomyosin cytoskeleton, and in particular, the activity state of myosin and the turnover of myosin could influence the polarization of these interfaces in a way that suggests that something that we think of as the final effect of these rho GTPase cascades could actually feed back to influence something upstream in the pathway itself. So in a way, what they suggested was that this sort of flow of myosin and its turnover at these interfaces could actually influence processes that occur upstream, so as to suggest that there is a self-organized biomechanical network that allows shape changes or changes in interface morphology that are associated with this process. So again, I just want to highlight the fact that in addition to influences that are coming from gene regulatory networks and regulators of the cytoskeleton, properties of the cytoskeleton itself, in particular, the activity state and turnover rates of myosin can feed back to influence steps upstream in this process. So if we saw till now that bending requires spatial restriction of contractility to the apical end of a cell. And in particular, it influences or it's dependent on medial actomyosin complexes. What we've seen with elongation is that also relies on apical actomyosin, but these actomyosin complexes are planar polarized along at the apical end, and these apical actomyosin complexes or the differential distribution of myosin in these is dependent on actomyosin flows. And the restriction of these, or the restriction of the direction of these flows is dependent not only on the gene regulatory networks that govern gene expression, but also on downstream effects such as myosin turnover and activity. So what I'm going to do now is move to a third process, and we will come back at the end of this to sort of discuss some common themes and some differences.
Maithreyi, some questions here.
At
What happens to the basal part while the apical parts are readjusting?
say again?
What happens to the basal junctions?
Basal structures.
Yeah, the junctions there.
Okay, I didn't talk about that, but essentially a large part of the work, all but one paper on germ band extension, has focused on events that are happening apically. Yusuke Toyama in Singapore has some years ago shown that there is a basal remodeling of junctions that actually precedes the apical remodeling of junctions. So it's fair to say that most work on apical constriction and this process has focused on the apical end, right? But we have to remember that cells are three-dimensional structures. And as Vijay may actually tell you, cells are three-dimensional structures that have to be packed on curved sheets. And therefore, thinking of them as cylinders and wedges is a bit simplistic, substantially simplistic. And some of you may have read that people have also discovered new cell shapes that are called cutoid that explain how you can pack cells on a curved sheet. I'm not going to talk about that, but so that's what's happening basally, and there's probably a lot more that's happening basally too. And that's true for apical constriction, it's true for dorsal closure, and we know that some of it is true for elongation.
So the other one was sort of a clarification, which is, does this germ band extension happen immediately after ventral furrow formation, or am I missing the point there? So does ventral furrow formation precede germ band extension?
What? Affect.
Proceed.
Yeah, so there's a tiny overlap between the beginning of germ band extension and the finishing of. There's a small overlap, temporal overlap.
Ah, I see. No, because I was wondering if, because you mentioned at some point that mechanical induction could, there could be a mechanical induction of the myosin polarization.
Yeah, so people have looked also at the effect of germ band effect on germ band extension of mutations in fog, for example, that influences both posterior midgut invagination and also ventral furrow formation. So as a way of addressing whether all of what we just described are solely due to the intrinsic changes that are happening within the cells of the germ band, or whether external influences such as simultaneously occurring morphogenetic movements that deform tissues could also contribute. And if you remember that first slide, one of the first things that appears to happen prior to these junction shrinkages is actually a stretching of the tissue, right? Without the intercalation. So one way of thinking about it is that stretch is a trigger for subsequent intercalation. So it's a bit like saying you could have uniaxial stretch. So if you took a sheet of cells in a dish or on a substrate and then stretch them uniaxially, we know that has effects on the organization or packing of that monolayer that grows on the cell. So one way of thinking about it is that deformation, for example, stretch induced by other morphogenetic movements that are overlapping in time, would serve as triggers for this. And then there have been studies where Matteo Rausi, who's going to be here, not this coming week, but I think the week after, studies where been looking at the effects of cauterization, which is a way by which you can physically separate two parts of a sheet or two different parts of the embryo without actually inflicting a wound. So you basically make scar tissue, let's say, and then looking at how the process or the cellular events are actually modified. I think he's going to talk about it. There's some mention of cell intercalation in one of his talks. So I think you should let him speak about it.
Sorry, just one last one. So this convergent extension and the multicellular rosette formation seem to be sort of symmetric in the x direction. Meaning this extension itself is sort of symmetric in the x direction. So I would have thought the tissue moves in both directions when extending in the embryo, but it moves predominantly along the one.
When you say both directions, what do you mean? You mean is it like
towards the anterior and the posterior, but it seems to move predominantly towards the posterior. So there must be something blocking the movement.
You mean, is it like so are you asking whether such intercalation events happen uniformly throughout along the anterior posterior axis of the germ band? Or no, so I didn't.
Okay, so that could be one way, I thought. So when we look at the movies, right, we see the germ band sort of shrink in the y-axis, but elongate in the x-axis. But it doesn't do so in the two directions equally, sort of, right? Or it moves up and forms a U along one direction, but not along the other.
Yeah, so you're basically asking why is it folded up like a U as opposed to sort of maybe being like a long thing that just stretches the embryo like that?
Yeah, so why does it move predominantly along the one direction?
Yeah, so I would say I don't know the answer to that question, but I would imagine it's because of other steric constraints in the embryo, perhaps. I don't know. I don't know.
So I was confused. So you said that stretching can act as a trigger between the two axes for the, you can say the interface, like we're telling which uh, orientation of the interface is getting myosin predominantly expressed enriched. So is it that the myosin being deposited there, the increase in contractility causes the tension, or is it the tension which causes the myosin to get polarized in that interface? So that's what I wanted to ask. And another question, if it's known, is
Wait. So you're asking about the myosin polarization
so
to those shrinking interfaces.
so does something chemical cause the myosin to come over there at the interface, and which causes the change in tension, or is it that there's a difference in tension which causes the myosin to come? Or one way to ask this would be: is it known that the myosin at all the different cells starts increasing at about the same point in time, which would be like there's some global thing that's causing it? Or is it like there's locally changes, and that is it like there's a synchronized like polarization of myosin at the AP interface, I think. One of the interfaces, I don't remember. So, or is it like there are local unsynchronized changes, which might be some chemical?
When you say local, how local do you mean? You mean at the level of individual interfaces?
Say that if you stretch something, that would be like a global effect in my mind. But if you have some chemical effects, you will see a change which is not that synchronized. Maybe that if you look at a longer time scale, you will synchronize. But you can tell the difference between a chemical effect and a pulling effect.
Yeah. So I guess. Okay, what I'm going to say is that it's not going to be just one or the other, right? It's going to be a combination of the two. Simply because even if you have to transduce something that's physical, you do that through chemistry in some way. So so when I said that stretch may be a trigger, I was thinking of it as let's say a predisposing condition that will then facilitate whatever chemical processes that need to happen at those interfaces to allow those remodeling events. So there is even prior to junction shrinkage, there is an AP-DV difference in the enrichment of myosin at these interfaces. As the junctions shrink, other things happen, but you could say that DV-AP axis polarization with respect to these proteins I showed you is genetically hardwired. There is already an asymmetry that's pre-patterned. It's not that all the DV interfaces are shrinking at the same time. So they shrink. I would say the tissue on the whole behaves stereotypically, but not all junctions are shrinking and elongating at the same time. There is some sort of patchiness or stochasticity in that process, and that must come from local conditions. Okay, so I'm now going to move to looking at a third process, which accomplishes what you might call the fusion of a sheet so that you create a continuous sheet. And what you see here is a process that happens towards the end of embryogenesis, so the third in sequence in Drosophila embryogenesis, during which these two striped sheets, I'll tell you why they're striped, actually move towards each other and establish continuity in the middle. So these two flanks, as I will call them, were actually separated from each other by this intervening tissue that had the bright big cells that someone asked me about. So what you will see is that this elliptical tissue contracts at the same time at which these two sheets of cells that are attached to it actually come and meet in the middle, right? So this happens. So I have to sort of remind you that what you're looking at looks nice and flat, but if I to take you back to the cylindrical view of the embryo that you cut in the middle, then essentially what you're seeing as this elliptical tissue will be a dome-shaped structure that's on the top, and what you're seeing as these striped flanks is this bit here, right? And so what you're looking at now is viewing this from above, right? So what you're seeing in the middle is this elliptical tissue, and what you're seeing on the two sides are these flanks. The reason so there are cells here too that are only faintly labeled, but the reason you're seeing stripes is because we've expressed GFP tagged to one of these segmentation genes so that you highlight columns of cells rather than the entire sheet. So what we're going to look at now is how do you accomplish the fusion of sheets, or how do you build a continuous sheet from two flanks that are separated from each other in the beginning. So one short answer to that question is that it requires events and behaviors that happen not only in these flanks, but also in this intervening tissue. And as you can see from this movie, the striking change in the morphology of this tissue is that the area of the ellipse that is occupied by that tissue reduces as a function of time, right? So you can think of this in the same way as you think of a wound. So if I make a cut in my skin, the two edges of the skin will gape, and then something will come and fill that space in the middle that will actually help bring the two edges together. But the cells themselves also need to do things to allow them to come close to each other. So what I'm going to do is to try and use these two tissues to build parallels with how each of these individual processes happens. And so what I'll describe to you first is what happens in this tissue that is attached to those flanks. You'll see that the ellipse is shrinking in size, but you'll also see that the cells that make up this ellipse are reducing in size. So this is the apical end of the cells. So just like what happened in the mesoderm during invagination, I can say that the apical ends of this tissue are actually constricting. What you will see, though, is that not all cells in this tissue are constricting in the same way. You'll see that the cells that are adjacent to the flanks that are coming closer actually shrink primarily in one direction. Here, you can see these cells here. You can also see that the cells in the middle are also sort of pulsatile, at least in the early stages. So their shapes are oscillating. And you'll see that there are some cells here that actually become very small, very fast, and leave the sheet. So you could say apical constriction within this tissue is not homogeneous. It's both patterned and there is a stochastic element in it, which is the fact that you have some cells whose position and timing of contraction and disappearance you can't predict a priori. So the reason this sort of fusion morphogenesis is important is because this is also how the neural tube forms. What happens in that elliptical tissue that I'm going to talk about is a bit like how the neural tube itself is generated. What happens to the flanks, like what happens to the tissues that are abutting the neural ectoderm that help cover up the neural tube after it has invaginated. And if you have failure in this process, then the neural tube stays open to the outside. And so what I just showed you in that movie is that if you look at apical constriction in this tissue, you can distinguish many different kinds of cell behavior. You can say that the cells in the center actually oscillate, their shapes oscillate in the early part of this process, but constrict quite rapidly at the later on in the process. Cells that are in the periphery constrict anisotropically, and then some cells constrict very rapidly and leave the sheet. So you could say there are 4 different modes of apical constriction in this tissue that contribute to its contraction. And if you look at some of these modes, and I'll focus only on one, because of its seeming resemblance to what I showed you during the invagination of the mesoderm, and that is pulsed constriction. And what you will see is that the cells are actually exhibiting changes in apical area that look like this, right? Which is different from the stepped pattern that I showed you in the mesoderm. What you're seeing in the insides of these cells are blobs of myosin that form and dissolve. And you'll see that from the time of the formation to the time that they dissolve, they've also moved a certain distance within these cells, right? So what is marking these cells on the outside is an adhesion molecule, simply to show you how the changes in myosin intensity correlate with the shape of the cell. So you can see that when the myosin blob is present, there's local constriction, and when it dissolves, there's expansion. So I'm primarily going to focus on this just to tell you that what pulsed constriction in the amnioserosa is associated with is again an apical and medial actomyosin complex that appears to move. I mean, in addition to forming and dissolving, it appears to move in a sort of random direction, right? It could move here, it could move there. What distinguishes this from the stochastic process that in which some cells constricted rapidly and disappeared from the sheet is that the myosin blobs in the nearest neighbors travel persistently and in a directional manner, but in the cells that are surrounding the cell that constricts, right? So I'll make one statement here, which is that if I look at pulsed constriction, I can say that these dynamic actomyosin complexes that form and dissolve appear to be randomly directed, whereas when a cell is constricting rapidly to exit the sheet, these complexes in the neighboring cells appear to move in a direct towards that cell, let's say for longer distances, let's say more persistently, but in the neighbors of these cells. For today, I will focus on these two just to show you some differences. And then, so what I was showing you is this here is pulsed constriction, and this here cell delamination. And what you can see, some blobs of myosin that are directed towards this delaminating cell. So one can begin to look at these pulsatile contractions, and then if you saw that field of cells that was with myosin, you would see that not all cells are not pulsing in phase. Myosin is not polarized to interfaces oriented along some embryonic axis. So you can begin to ask if there's some sort of communication between cells that governs this pulsatility. And one can begin to ask that question by finding ways by which you can either perturb single cells in a sheet or use lasers to create changes in shape and geometry. And by doing experiments of this kind, what you can uncover is that if you perturb a single cell within this sheet, right, and what you're doing here is either activating contractility in a single cell or reducing it by playing around with these rho GTPases that switch, you can see that you affect not only the pattern of pulsatility in the cell that expresses it, which typically shows a dampening of these oscillations, but you also see a dampening of these shape fluctuations in either just the nearest neighbors or in the nearest neighbors and a circle of neighbors around it. So what this tells you is that although these pulsations look like they're not in phase, a cell in a sheet is sensitive to the state of contractility that could be spatial, temporal, whatever, in its neighboring cells. And this is also reflected in the amplitude of the pulse, which is reduced at least in the nearest neighbors in both of these cases. What's perhaps a little more striking and somewhat unexpected is you can take a single cell in which you've activated contractility, or you've used a laser to create those changes in geometry I showed you, and then see that in both of these kinds of perturbations, you can now redirect myosin towards the interfaces of that cell that you just constricted by activating Rho, or you caused to expand and constrict by using a laser. So what this essentially tells you is that in the sheet of cells where you have pulsed contractility, a cell is sensitive and responsive to the state of contractility in its neighbors. And this suggests that although things might look random, if one could map things like stresses within the sheet at distinct interfaces, one might be able to correlate the pattern of pulsatility with the dynamics of stresses operating at interface. So this tells you that these cells are actually mechanically coupled, and this mechanical coupling might influence what its neighbors are doing. So you were asking about what the point of having ratcheted constrictions is, and I did say in relation to ventral furrow formation is that one possibility is that it allows a tissue to sort of sense what its neighbors are doing and respond to it in a way that maintains stability of both shape, dynamics, and integrity of the sheet. Okay, so now just to ask this question that we were asking earlier on with germband extension of whether you actually need the neighboring cells for something, right? One can now look at what happens if you use a laser to make an ablation in a cell.
Maithreyi, in
a cell that's wild type.
In the last video.
Let me just switch this off. Just 1 second.
The previous slide. The time scales for these videos are way too different. So one of those is going for hundreds of minutes, seconds, and this is.
Yeah, this one?
Yeah, this is going for like 10, 15 minutes, and this is just few seconds.
What's the question?
The time scale are that different for both processes?
Again?
The time scales are very different for both processes. The videos.
Yeah, the time scales are very different.
Like
so
tens of orders.
No, so the time scales are very different. So all of this happens within 10 minutes.
Yeah.
So here we've used a laser to make a perturbation that's largely cytosolic, that's like an ablation directed at an interface, allows you to see the expansion of the cell as a result of the release of pre-stress in the cell. So the one thing that this is accompanied by is the expansion of the cell you ablated, which is then followed by constriction of the cell and its extrusion. Here, what we've done is to simply activate contractility in a single cell, right? Of course, the time scales are very different here. All I'm trying to say is that the effect it has, so both of these perturbations can redirect actomyosin organization in the neighbors. That's all I'm saying. Your question was that the time scales are different? Yeah. So if I look at a cell that's also going to sort of extrude from the sheet on its own without us using a laser to induce it, the timescales there are also longer, right? In the sense that from the time that it begins to rapidly constrict to the time it exits the sheet is much longer than what happens here. But all I'm trying to say is that in both cases, you can redirect the myosin in the neighboring cell. So myosin that was initially randomly oriented during this pulsed constriction process can now be made directional, which therefore suggests that altering the tension at interfaces could be a cue that dictates where myosin goes. And so if you could, it just goes back to the point I was trying to make. So if we could map the stresses, and if we can't map the stresses, we can at least map the strains associated with each interface by looking at their deformation. The question is, we might be able to build a correlation between the movement or the direction of myosin movement and a stress anisotropy in the sheet. And what I showed you, or was going to show you in this next slide, was that in the same kind of experiment that involves laser ablation, if you ablate a cell that's normal, that's surrounded by neighbors that cannot generate forces by actomyosin contractility, what you will see is that the cell that was ablated begins to expand, then it constricts, but it's unable to complete this constriction, then starts to expand again. So it does not extrude from the sheet. So what this sort of allows us to think about is that even events that might involve rapid constriction and exit from the sheet are dependent on forces that are generated in the neighbors to facilitate them to leave the sheet, right? And I showed you earlier on that for that process of constriction and extrusion, you also see that there are directed flows of myosin in the neighbors. So what I'm trying to just use this to say is that in this sheet in which you have apical constriction, every cell is in tune with what at least its nearest neighbors are doing. And the nearest neighbors do matter and can modulate aspects of contractility in the neighbors. Okay, so that was the first part of what I wanted to tell you. And that basically tells you, that basically also allows me to compare what we discussed during ventral furrow formation. The similarity is that for the invagination of the mesoderm and the contraction of the amnioserosa, you need apical constriction. Whereas you saw that stabilization and contraction, so stepladder type pattern in the ventral furrow. In the amnioserosa, you have two phases. One in which you have very little net reduction and the areas kind of oscillate like that, right? It's followed by a phase where you have very little oscillation and the area reduces more drastically. The second is that in this model, you have a heterogeneity in the apical, in apical constriction, and you can link these heterogeneities to differences in the dynamics of myosin. The third, it actually allows you to see in what way local influences from neighbors might act to determine contractility in the sheet, in addition to global effects. What I've not told you anything about this apical constriction is what are the gene regulatory networks that govern it. And I will say that in one of the early slides on dorsoventral patterning, I showed you that the mesoderm was formed here, where you had high dorsal nuclear gradient. The amnioserosa was formed at the opposite end, where you have low nuclear dorsal, right? This in turn, this low nuclear dorsal has effects that effects on signaling pathways that in turn govern the transcription of genes that specify the amnioserosa and also regulate its behavior. We don't yet know what ligands on the membrane, what ligands bind to what receptors on the membrane to initiate apical constriction in this tissue. But what we do know is that the downstream effects involve the activation of Rho GTPases. And in particular, what we also know is that between this phase and this phase, the dynamics of a regulator of active contractility behaves differently. We still don't completely understand how these temporal differences in the activation of Rho between these two phases are governed. So I'll just spend few slides to draw a parallel to the second part of the second process I spoke about, which is germ band extension. And all I want to use this to say is if you look at how the two sheets that were striped in the first movie I showed you actually meet up in the middle. If you watch the way these contacts are formed, what you will see is that initially the contacts sort of form head on like this. So that if you look at the morphology of the interfaces, at the initial points of contact, they will be like this, right? If I had the two stripes. So this set of contacts is kind of horizontal. What happens to those contacts progressively, as you will see here, those interfaces that just formed after fusion will become a little floppy, right? So they might look like this, or they might look like this, right? And so what this will translate to are interfaces that look like this, right? So in some sense, what this resembles is the opposite of what I showed you here, opposite in sequence at least of what I showed you here. As in, you start off with a set of morphologies like this, and you remodel them to morphologies that look like this. So what I'd like to sort of say is that if you look at epithelial fusion, you have the bed tissue that's doing something similar to what happens in ventral furrow formation, and the flank that fuses by mechanisms that may not be very different from what happens when you're doing junction remodeling for tissue deformation, except that we're not actually shrinking the junctions in this case, we're expanding them. Create a nice linear seam that becomes interlocked. And I will just end with one slide that says that even for this sort of junction remodeling event, you have a very spatially regulated spatially regulated remodeling of junctions. By that I mean only the junctions that are actually forming upon fusion are remodeling in ways that are characterized by a reduction in the levels of actomyosin and an increase in the amount of bazooka. When I say increase, I actually mean new recruitment of bazooka, because what happens fairly soon after these contacts are formed is that you begin to recruit this polarity protein bazooka to these interfaces. And you are concomitantly clearing actomyosin out of these interfaces. So in a way, we think that this formation of a continuous sheet actually requires junction remodeling that's similar to what you see in the junction growth phase of cell intercalation. So you could think of epithelial fusion as requiring apical constriction in one tissue and very locally regulated junction remodeling to both facilitate the formation of a hexagonal packing geometry, but also potentially to confer some stability on these newly formed junctions. You might imagine that if cells are just stuck to each other like this, then small stresses might operate on these weak links to disrupt integrity, whereas if you have a morphology like this, if a fractal line, it's not going to cause massive disruption to integrity. So I'm going to leave you with that, and in the next lecture, I'm going to cover complex morphogenetic movements using processes in zebrafish and the mouse to think of more three-dimensional, more strikingly three-dimensional and multi-tissue events. And spend a little time at the end discussing how much pattern or form you can generate in a dish.